It's my great pleasure to welcome you all here at Indiana University, but also online. You see here the link to the website. Please feel free to go there. It has the full agenda and also has the set of participants. As you see, we have a hashtag. Please do tweet and make up sure that other people learn about this event. As you might have heard, all of the presentations are recorded, also streamed, and all the recordings and slides will be made available after the event. This event is organized with Jeffrey Spraggins and Sanjay, Jain, the local, and it's wonderful to really do this collaboration. It was very efficient collaboration. I am looking forward to do more of these. And there are many local organizers, two of which are listed here, Lisa Record and Matt, which some of you might have been in contact with. There are many others which are taking good care of the technology here and also will be answering questions that come in via the chat. The agenda for today and all these times are Eastern Daylight Time is to have a brief welcome by Richard Conroy from NIH, which just happened, also by us organizers. We have then brief introductions by participants. We have a brief overview, 30 minutes, by us three organizers. Then we have 4 sessions, one on metadata, one on ontologies, one on anatomical mapping, and one on the CCF user interfaces. And then for those which manage to come here in person to Bloomington, Indiana, we will all meet at a local restaurant to have even more discussions. Going over to the brief introductions by participants, maybe let me just start with Jeffrey and Sanjay. They might like to introduce themselves and welcome you all as well. Jeffrey, if you want to start.
Sure.
Yeah, you're already unmuted. Wonderful.
No worries. Yeah, my name is Jeff Spraggins. I'm a research assistant professor here at Vanderbilt University and one of the PIs on the Vanderbilt TMC project. And our focus is on developing integrated molecular imaging technologies. And in our case, we're tasked with applying that to developing 3-dimensional molecular atlases of human kidney.
Sanjay.
Hi, good morning. This is Sanjay Jain from Washington University, St. Louis. And I'm part of a tissue mapping center with UCSD for the kidney project. And our center is actually called CoolMap. It also has lung mapping center with Jim Haggard, Shin Sun, and Gloria Prague-Huber, and Peter Kravchenko. So, and our interest from the WashU side is to really do the RNA as well as chromatin accessibility studies and 3-dimensional mapping of the kidney. Thanks.
Thank you both. Again, wonderful to collaborate. Coming here to this room, let's go from right to the left. So Lisa, if you want to briefly introduce yourself.
Okay. I'm Lisa Record. I'm here with the NCIU team at Indiana. And my focus on the project is primarily with user studies and project management.
Jim?
Hi, I'm Jim Sluker. I'm part of the NCIU team, and I'm responsible for the ontologies. I'm a Stephen writer with the National Library of Medicine. I'm responsible for research on the biomedical and biomedical terminologies and ontologies.
I'm Börner from the University of Auckland. With Peter Hunter, I am co-PI on the MapCore grant for SPARC. SPARC is focused on creating a neuroanatomical map across the body, across species, and I'm developing a theory and knowledge presentation for anatomical connectivity.
Leonard?
Leonard Cross, UX, UI design for the ontology CTF portion of the project.
And Katy Börner, I serve as the lead PI for the NCIU component. Matt?
Hi, Matthew Martindale. I'm with the NCIU team. I'm a local organizer, a center assistant at CNS.
Ellen?
I'm with MCIU. I am a biologist. I am dealing with anatomy and molecular biology and ontologies.
Thank you. Paul?
Hi, Paul Macklin. I'm in MCIU working with Katy on the ontologies development. She's a mathematician who works a lot in computational modeling, cancer, and other fields.
And now I have to rely on Ellen and who she sees and who she can unmute. So we would love to have everybody introduce themselves briefly.
Okay, Jessica Page.
So I see Zorina. Zorina could introduce herself.
Can you hear me?
Yeah, I can hear you.
So I am Zorina Gallus. I'm at the Hartlange-Bland Institute, NIH. I'm part of the HapMap coordinating team, I guess. I started my life in science as a physicist, and I trained all the way to pathology and vascular medicine. So I'll try to give you a presentation that will try to incorporate everything I learned in the past 30 years, and I'll try to zip through 10 minutes, but I'm hoping to start the conversation. So thank you so much for having me.
Thank you for being here. Oliver He, can you unmute yourself, or maybe Ellen can do it?
Yes, I'm Oliver He. Let me see if should I start a video. Yeah, I'm Oliver He from University of Michigan Medical School. I'm now working on ontology work for the KPMP project. Thank you.
Thank you. And that's all I see in my interface. I'm not sure if we see non-presenters. All right, going through the slides and in the interest of time, thank you for these introductions. And again, we are now going over to the HapMap overview by organizers. Before I go there, I wanted to make sure that you saw that we started at Challenges and Opportunities Google Doc. So if you go to that tinyurl.com/ccfws-feedback, you can see in real time how everybody is adding challenges and opportunities as they relate to metadata, ontologies, mapping and numerical construction, and user interfaces. We also added a space for references and for links, and the current information that you see here comes from the registration information. So many workshop participants provided extensive materials via the registration form, but we also took the liberty to add some from the MCIU team. So again, that tiny URL, if somebody could also post that to the chat link, that would be great. And maybe we have to repost for those which join later because they wouldn't be able to see it. So coming over to HapMap, it's a very exciting project. It's a team of 140 plus experts now. And the vision is to catalyze the development of an open global framework for comprehensively mapping the healthy human body, adult body that is, at a cellular resolution. Here's also the link to the Commons Fund website, which has even more information. The goals, specific goals, are to accelerate the development of the next-gen tools and techniques for constructing high-resolution spatial tissue maps, to generate foundational 3D tissue maps, to establish an open data platform, to coordinate and collaborate with other funding agencies, programs, and the biomedical research community at large. And we were extremely happy to see all the many different participants coming in from non-HapMap projects, and also having here the National Library of Medicine presented, and Spark and some other teams that will come here physically. It's just wonderful to see. And I think this is larger than life effort. So I think we want these collaborations. And number 5, support projects that demonstrate the value of these resources developed by HapMap. This is part of the team you see here at the recent all hands meeting. HapMap is now 7 months old, so we're a very, very young project. We have a consortium website, which is the hapmapconsortium.org website. Again, feel free to go there. There's also first data available from this site. If you look at HapMap, you see there is a set of tissue mapping centers which were funded via U54 mechanisms, 5 of them. So Sanjay and Jeff, my 2 co-organizers, they both come from 2 of them. There are 3 more. Then there is the HIVE, which does the technical core development of infrastructure tools and mapping. Then there is a collaboration unit, which is part of the HIVE, but also serves the entire research community and the entire HapMap consortium. And then, as you also see, there are transformative technology development projects funded. and there will be rapid technology implementation projects and demonstration projects. I took the liberty to apply our science mapping technologies to the HubMap effort itself. Here you get to see who got how much money. That almost tells you how much can be done by each team. So in the legend, you see that the area size actually gives you the total cost by the component. And you see that some of them have more years outwards, some have a lot of money in little time. So it's interesting for us also all to understand how this is currently set up. You might also be wondering where we are geospatially. Here is a map of the US. Here's a map of the US with overlays of where the lead PIs are and then their teams close by to them. You see that there's some long-range collaborations which are made. And then the lists on the lower right of the map, those are the European counterparts and partners. We did not manage to list all the NIH program offices. There are many. They are just beyond what we can do there. It's really a long list, and it's great to see the buy-in from so many different institutes at NIH. If you are interested in how we organize ourselves, this is from March this year. I took the roster information, and what you see here is the working groups. We have 5 major working groups, from communications and engagement to tools and models, data science, tissue technology, and data collection, and also policies and who is in which. So you, for instance, get to see that Ajay Pillai is on 3 of the working groups. Jonathan Silverstein, who is going to be here in person and very soon. He is also on 3, so some are very active in multiple working groups, making sure that knowledge flows from one to the next. But you also get to see who is mostly focusing on one of these. Now, with the planned data release for summer 2020, we have now a set of new working groups, and so this graph needs to already be updated. If you want to know how HubMap relates to some other efforts, and I really apologize for the RBK and KPMP and other efforts that are not yet captured here, but those which are captured here, you will see that, for instance, Michael Snyder connects us to HCAM, and Kun Zhang connects us to the BRAIN effort. And if you would map not just the PIs, but also co-PIs and senior personnel and maybe even postdocs, you would see even more connections between those different efforts. You might have seen that there is now a markup paper up on xArchive, which explains the HubMap effort in more detail, from tissue collection to different assays and analysis to data compilation to map generation to data storage and dissemination, and how these efforts are supported by these 3 different TDDs, RTIs, TMCs, and the Hive components. You might also have seen that HubMap is special and that it cares about 10 organs currently, and that's a challenge, it's a true challenge. You might also have seen that we care about many different assays, and we will get into details later in the presentations on how many assays we actually have here. What you see in the lower part of this figure is the usage of landmarks to do image registration, and then based on registered images do feature extraction also in 3D, and then coming over to a common coordinate framework, which our team and Rahul's team are responsible for. If you are interested to poke into data, there is first data available served by CMU, the Pittsburgh Supercomputing Center, and the University of Pittsburgh via the HubMap Consortium website. So feel free to check that out. I will now give over to Sanjay and Jeff. They are going to take on, they're going to use their computers to present their part of the story. Thank you all.
Yeah, thanks, Katy. I'm going to share my screen soon.
You will need to stop sharing. I don't know how to do it. Which computer would do that?
Just
Not my
stop
computer.
share, whoever's in.
Yeah, just with the mouse on the and the keyboard next to it.
No.
Can you see my screen?
Not yet.
Okay.
I can see you. That's a good sign.
Should be coming soon.
There we go.
There we go.
Okay. So, hi, good morning. Sanjay Jain from WashU. And what I'll give is a little very high-level view of some of the, like basically summarizing what our project is, our team, and some challenges with the kidney regarding common coordinates, and then maybe some areas where we can resolve this in terms of harmonizing with the other tissue mapping centers and TTD, technology development centers. So, this is basically our team. Our project is called CoolMap, which is where contact PI is Kun Zhang, and I'm leading the WashU side for the bladder, urinary tract, and the kidney mapping project. And we have a local pathologist, Joe Gout, at WashU, who is my co-investigator. And then Jim Haggard, Shen Sun, and Gloria Pryhover are on the lung mapping project. And Peter Krachenko is from Harvard University, who is in charge of the data analysis and coordination and distribution to the hive. And Blue Lake is in Kun Zhang's team, who is working primarily with us on the kidney project in analysis of the data and technology implementation. So this is the project overview, and the top is the cartoon of lung, and here is kidney and the lower urinary tract. The kidney is organized in these lobes, and it has cortex and the medulla, and then the urine is taken down from the ureter into the bladder. So our project is to basically use three main technologies. One is called Dartfish, which is a multiplexed 3D in situ hybridization technology. And the second technology is single nucleus sequencing using drop-seq or split-seq, is to uncover the cellular diversity in these regions. And the third technology is SCTHSC, which is a chromatin accessibility study. And then there's another technology in development, which integrates both the single nucleus sequencing as well as chromatin accessibility studies. And these technologies are being actually implemented from Kun Zhang's lab. So the idea is data generated from each of these technologies are integrated into these sort of mini atlases, if you will, which create basically organ-specific spatial maps of all the cell types, their spatial distribution, their reference transcriptome, reference chromatin accessibility map, and selected proteins in the 3D space. So this is just an overview of our technologies. So what I would do in this talk is basically give you some of the challenges in using common coordinates in the kidney, a very, very high level summary of that, and maybe some approaches how we minimize these challenges, and maybe and a little overview of our workflow for the bookmap project. So just give you one example of kidney anatomy. So one of the challenges in kidneys, and it comes really from the development perspective, how kidney develops and begins in the lower pelvis and comes into the abdomen in the retroperitoneal space, is one of the things which happens is the variations in kidney vasculature. So here are two kidneys on either side. This is the aorta, and this is the main renal artery. But you can appreciate is that these arrows are pointing to these accessory vessels that are coming into the kidney from one side here, and there's another one over here. The second example over here shows on the right side over here, both of these, two accessory vessels into this kidney, and another one on this kidney. And here's another example where only one sided there's this variation. And here's the contralateral kidney with another variation. So this is just example to point out that inherent developmental anatomical variations sort of make this problem of unifying a common concept of using vasculature to map spatial locations in the kidney. Not only that, the way kidneys are placed in the body, external organs around it can also impact how vasculature can be affected. For example, over here on the right side, this is an outline of the spleen, and you can see how it's compressing this upper part of the kidney and distorting this vasculature, which will affect on how the neighboring microstructure so will vary. So this is example from the vasculature, and here is another example of variation in the collecting system. So collecting system in the kidney is one which actually is responsible for development of the adult kidney. And what happens over here is the collecting system, this is the pelvis and the ureter, it is organized into many different lobes, and it branches reiteratively to give a very repetitive structure. But then there are variations in the collecting system, as shown over here. If I draw a line from the upper to the lower pole, on the right side, you can see these branchings over here. But on this side, if you see on the left side of the line, the main branches have happened before this on the left side of this line. So, and on the right side is just an example of many different variations can happen in this collecting system. So this also sort of poses challenges in terms of assigning uniformly in a timely manner the anatomy and common landmark nomenclature for these. So so the way we can deal with this, which I'll probably go in the next few slides. So one of the things is to really have good quality control and standardization to have effective coordinate system. So, and this sort of exercise starts right from the subject all the way going to the tissue and to the imaging and omics analysis and even data deposition. And what I list over here are different parameters that we can standardize and quality control to harmonize a lot of the data output that will occur from these efforts. For example, pre-analytical parameters, the metadata, the clinical, the procurement of the tissue, storage, shipping, and tissue collection, the histological authentication, anatomical mapping as best as we can, the processing pipeline, and in the omics level, how we define the cell types, agreement on that, reproducibility and validation, and even instrumentations and quality control cutoffs. And at the data deposition level, from the Hive of and data coordinating center, there also have to be standardization so that data can be interpreted across sites, across organs, or across tissues and cell types. Is this analysis approach that can be harmonized, softwares that can be harmonized, ontologies and visualization. And some of these topics, speakers later in today's workshop will allude to and give a, and I'm very interested to see all the different points of view when in these talks. So one of the ways for the kidney is the gross registration using Porter lines. So this is an approach that our surgeons use, especially for the tumors, but it sort of serves as a little bit a uniform level at in the gross kidney. So these are called, so the kidney has this, this bean-shaped structure, and this is a little upper lip and the lower lip. So if you draw the horizontal lines in this coronal plane, one way to simplify divide is this, and the upper pole and the lower pole. And then this would be the middle pole. So this is one way to at least donate at some level, denotes at some level where you are taking your tissues from. Of course, there can be higher granularity on this, but remember the variability I showed, that could cause problems. But at least this sort of serves some uniformity. And here's an example from a specimen from a deceased donor. Here's the kidney you can see, and this is the ureter over here. And if I cut it open over here, this kidney in the long axis, you can see is if you take tissue from this lobe over here, this is the upper lobe, this is the lower lobe. So this is a very high level gross registration of a deceased kidney. And here's an example of a nephrectomy specimen from a kidney tumor. Here's a tumor. This is the lower pole over here. This is the line. And this is where we took the kidney from. So we can capture these images. We can know where the posterior and anterior aspect of the kidney is. We can measure the depth of this kidney and then register how far deep from the posterior or anterior aspect we have taken tissue from. So this gives some level of gross registration in the kidney. The second level over here is describes more of the workflow at the tissue processing level. This cartoon just illustrates the different areas that we will, over the 4 years, will be a little ambitious, but we would plan to, map in the hub map. So this is a when we take the tissue, there are many different ways we can process it, and we standardize these protocols. such as formalin fixation, fresh frozen OCT, fixed frozen OCT. And the idea is that using these standardized different processing protocols, a lot of technologies will use one or more of these things. So even if we use one of these methods for our technology, the adjacent tissue or adjacent tissue process with different way can be used in other technologies in HubMap. And here I'm showing you an example of a frozen OCT. So the way we do this, we use this frozen OCT for 3 different technologies in our mapping center, and adjacent tissue can be used for registration of the histological composition of the tissue that we took. So then we can have a histological reference with the microanatomy of the kidney. We can have a cell type state reference from chromatin accessibility and single nucleus RNA sequencing, and we can have structure region reference when we do the 3D mapping. So this is at least our plan, and we will continue to further crystallize this as we get more data, and this will be a process in evolution. This is just give you an example of microscopic structures. So the kidney is a repetitive unit made of nephrons. This is just showing you a photograph of the specimen from the cortex. This is the capsule. This is called the corticomedullary junction of the kidney, and beyond this is the medulla, which is shown over here. It has an outer part and inner part, and these are just the histological images from some of these areas. And this is the ureter. Here's the cross-section of the ureter at a high power. So this sort of tells us what the composition of the tissue is that we are dissociating to create single nucleus sequencing maps, and also which can also help us identify structures we can focus on for our 3-dimensional mapping. So here's an example that a study we did in the Kidney Precision Medicine project. This is a result from human kidney from 15 individuals, about 18,000 different nuclei, and this is the structure of nephron. So by adopting standards of regions and segments of the kidney that folks can use in a uniform manner and cell types, one could actually have some harmonization of what these clusters are mapping to. So this is a way to standardize segment names, cell types, and markers, which gives us cell quality control, as well as ability to go into annotating these clusters into different regions that we identified from the histology. So this is another way to minimize some of the variation and by adopting some standard terms. So the Vanderbilt University Medical Center team is also doing kidney, and you will hear shortly from Jeff Spraggins. And because we both are working on kidneys, there's a way that we are now actually been working on to harmonize our different technologies at different levels. So there's different kinds of metadata that we have extensively shared, and also been in communication with other centers and Hive to propose some of the common elements that both of technologies can use. Anatomical coordinates that I just showed you an example, the structures, the regions, the cell types, and the markers, with keeping in mind that we have to apply these standards for quality control in many of these variables. So there's been significant progress in them, but there's also some challenges, which hopefully we will get better at addressing. So, with that, I think this is my last slide. So I will hand it over to Jeff Spraggins for Vanderbilt.
Thank you, Sanjay.
Okay, Sanjay, you need to, at the top of your screen, there should be a bar that's green and red, and you need to relinquish control.
I think I could just take it.
Yeah, I think you can hit share. You get it? Yeah, I see your slides.
Okay, yep.
All right, let's see. You all see my slideshow?
Yep.
Okay, and Katy, just to confirm, we have 30 minutes, right, until the next session?
We have till 10, yeah. I think the introduction by participants was faster than we expected, so you have more time than we thought we would have.
I just dropped in some more slides because I saw the time, so we'll see.
Yeah. Wonderful.
Okay, so my name is Jeff Spraggins. As I said in my introduction, I'm part of the Tissue Mapping Center at Vanderbilt. We call ourselves the Biomolecular Multimodal Imaging Center. And the team, like all the TMCs, is effectively broken into three cores. The coordination core, which is led by Richard and I, the two PIs on this project, Danielle Gutierrez, our project manager, and Diane, our administrator. We also have, our focus is on kidneys. So we have a kidney organ specific project that is led up by Mark DeCastiker, Ray Harris, and Agnes Fogo, who are our resident kidney experts and pathologists. And then we have a data analysis core that I'm just highlighting some of the folks here. Ralph van der Plas leads up this team from TU Delft. His focus, or his specialty is multimodal image integration through image fusion. Ken Lau, who works on segmentation, and then Heath Patterson, who has done a lot of our registration development. Now, the Vanderbilt Tissue Mapping Center, the goal of our center is to effectively integrate three primary data sources, the first of which is high content imaging, or which we refer to as imaging mass spectrometry, which I'll talk more about in a second. High resolution, high spatial resolution imaging. For us, that means microscopy in general. And then multi-omics, and again, for our center, that is primarily metabolomics, lipidomics, and proteomics. And so our mission is to build a platform that integrates these modalities into a common multimodal molecular imaging platform. And we take advantage of three capabilities that we have here at Vanderbilt: our state-of-the-art mass spec-based molecular imaging, so imaging mass spectrometry, which has been pioneered by our team here at Vanderbilt. A world-class clinical environment that gives us access to tissues on a weekly basis and kidney pathologists through the Vanderbilt University Medical Center. And then some unique capabilities that we have in terms of data integration and analysis, as I said, based in both TU Delft and here at Vanderbilt. Now, overall, the way that we are constructing our pipeline is integrating imaging mass spectrometry, multiplexed immunofluorescence microscopy, autofluorescence microscopy, and then, as I said, spatially targeted multiomics, and then finally stain microscopy. So we're building all of these modalities into a single workflow. This is just a cartoonish representation of how this goes, but effectively we collect tissue, whether that's from unused donor tissues or through kidney resections. We then divide that tissue into either fresh frozen or fixed for storage. We primarily use fresh frozen tissue for all of our modalities. And then each of our modalities is inherently two-dimensional in nature. So to perform the experiment in 3D, we're effectively doing sequential sectioning through the tissue volume, generating many two-dimensional slices and performing our experiments on each two-dimensional slice independently and then registering and integrating after the fact. So we effectively end up with this sandwich of tissue that each layer is going to run down one of two pipelines. Either first of all, we collect autofluorescence everywhere, and I can talk about why that is, but it helps us with registration. It gives us a common modality to do all of our registration and integration later on. And then on one tissue, we'll do imaging mass spec followed by PS stain, and on the next tissue, we'll do multiplexed immunofluorescence, and we'll effectively cycle between those two pipelines throughout the volume of tissue. Now, as I said, the four modalities that we're focusing on are listed here. Our imaging mass spectrometry pipeline focuses on metabolite imaging, lipid imaging, and protein imaging. We're using our multiplexed immunofluorescence microscopy data to effectively label cell types within our tissue so that we can correlate our imaging mass spec data to cell types. And then, as I said, our autofluorescence is just a common imaging modality that allows us to register our data more accurately. And then our multi-omics, although we're primarily for HubMap, we're doing metabolomics and proteomics. We have done RNA-seq in the past, and this is primarily to give us deep molecular coverage and help us with identification. Now, most of you are probably familiar with the majority of those technologies. I just want to highlight the imaging mass spectrometry, which is really unique to our center and maybe unique to many of you. But our version of imaging mass spectrometry uses what's called MALDI, or matrix-assisted laser desorption ionization. So we apply a MALDI matrix to a tissue section. This is just mounted on a glass slide like any other microscopy-based approach. We apply this matrix, then we fire the laser. So this is a laser-based ionization strategy. So wherever the laser hits on the tissue, we generate ions. We then collect and detect those ions in a mass spectrometer. So we effectively get a full mass spectrum at every pixel location within our tissue slice. We can then select any of those ions and integrate its intensity over two-dimensional space to generate an ion image. Once we've identified those, we can say what lipid or metabolite or protein that ion is associated with. I'm not going to go into a lot of detail, but just to give a couple of examples, as I jump ahead, of some of the data we've collected for HubMap so far. This data is available and has been shared with the Hive. I think Katy provided that link to where you can see some of the HubMap data. But this is just a feel for the kind of multimodal data that we can collect with our pipeline. So basically, if you look at this white line, everything to the left of this white line is collected on a single piece of tissue. So we first collect autofluorescence and then imaging mass spectrometry. And then the imaging mass spectrometry does not damage the tissue, so we can collect PAS stain microscopy images following that. And so effectively, again, the autofluorescence gives us a common modality. The imaging mass spec, I'm just showing three distributions here of hundreds to thousands of ions that are detected, but these are three lipids that we've detected and localized within the kidney tissue, and then again, we can do the PAS stain. On a serial tissue section, we do autofluorescence, although I'm not showing it here because it's redundant, and then we also follow that up with multiplexed immunofluorescence, which I'm just showing a 3-channel experiment here on the right. And this allows us to then eventually correlate specific cell types to our imaging mass spectrometry data. As I mentioned, the goal of HubMap and the goal of our center, obviously, is to do this in 3 dimensions. And this is just a brief example where we've done this as just a test run as part of HubMap. So this is a 3-dimensional molecular imaging data set collected using imaging mass spectrometry. So these are 32 serial sections. Again, the registration is all done with autofluorescence, and then we can project our imaging mass spec data into these volumes. And these are just showing the distributions of a few lipids here within the cortex of the kidney. And I think this data set makes a nice point of how complex these data are. Below here you see a spectrum, and I've just highlighted a couple of the peaks that we've selected to show the volumes of. But in this case, depending on the instrument that you run this on, you collect hundreds to thousands of ions, and each of those can be represented as an image. Now, when we think about multimodal imaging and how we integrate data overall and how that relates to the common coordinate framework, we think of, we have a whole set of modalities that we're collecting as part of HubMap and some that aren't necessarily being collected as part of HubMap right now. But the goal is to connect these in some intelligent ways so that we can mine the data more deeply. The problem is, the challenge that we're trying to overcome here is that very often in multimodal imaging studies, it's easy to effectively silo your data and limit the amount of crosstalk between these different modalities, which you can still do manual interpretation, but it's going to limit the amount of information you can glean from any of the modalities separately. Now, just to make the point, one of the things that we are working on so as I said, in our TMC, we're doing imaging mass spec, stain microscopy, and tag microscopy, but we're also working with the UCSD team in WashU to start to integrate their RNA-based imaging dartfish modality with our imaging mass spectrometry data. So that's something we're working on here in year 2 of the project. Now, from a HubMap perspective, I kind of repurposed this figure here in the top left because I think it ultimately applies to HubMap as well. One of the goals of HubMap is to really fully integrate this data through a common coordinate framework. So how do we integrate our high content imaging with the variety of high resolution imaging modalities that are being collected by other TMCs and the single cell multi-omics types of analysis? And so the question that the CFC or the CCF is really trying to answer here is, how do we connect this? And when we think about connecting data, we effectively have 3 hooks. The goal is to put the data on a common footing. And the hooks that you have are content, so you can look at the same molecules across multiple modalities, and that makes data integration pretty straightforward, but it'll limit the number of things that you can look at. you can think about integrating data in terms of time course studies and looking at how molecules modulate together. Although that's not really appropriate for our types of studies in HubMap, because we're looking primarily at normal tissue and we don't have the benefit of doing some sort of time course study. And then finally, and really most relevant to what we're doing here, is using space as a way to tie data sets together and tie information content together. And so it's really through an effective CCF that is going to enable this kind of cross-modality data integration, which will open up the ability to mine our data more deeply and unlock the full potential of the various technologies that we're collecting via the various tissue mapping centers. Now, because I have a little bit more time, I wanted to talk a little bit more about this and how we approach this kind of spatial linking of data, and just show an example of what we've done with Ralph Rondevos's team looking at data-driven image fusion. And so this is really going beyond simple registration. So if we have basically two modalities that we're trying to connect, let's say microscopy and imaging mass spectrometry, and this is a very rough imaging mass spec experiment, purposely chosen that way. And the first step that most people do is just registration. So they're just going to overlay that data together and use some manual interpretation to say, okay, that looks like it localizes to a specific cell type, in this case, the cerebellum of the brain. But through image fusion and some of the tools that we're building, we want to create models that allow us to make predictions and connections between the two modalities that really unlock full mining of cross-modality information. And this allows us to do things like predict a higher spatial resolution or predict to other areas of the tissue, which in the case of HubMap would be really important, as different tissue mapping centers would be collecting data from different regions of the kidney. We can start to make connections across those separate regions. And the way that we do this is through basically multivariate linear regression. We have a number of variables on the mass spec side. These are just ions, or in this case, lipids. And then as a very simple example, if we think about the microscopy on the right, these are just color channels. And we effectively tell the computer, okay, go look for all of the connections between these two data sets. And over time, the computer will start to find the weaker and stronger connections between the different variables and start to build a cross-modality model. And it's really this model which is going to allow us to connect the two data sets and start to interrogate more deeply. And it's this model that is the common coordinate framework is what's so critical for being able to link data sets in a similar way. Now, just an example of what this can do. This is an example here on the top where we've collected, on one platform we've collected, this is a higher performing platform, so it takes longer, but it gives us more spectral data. We've collected the data on just half of a brain section here. So on the left half, we haven't collected any data, and it's fairly low resolution. So it's 100 micron spatial resolution. I've just zoomed in on a bit here. And then on a different platform that's higher throughput, but less spectral information, we've collected the whole brain at 30 micron spatial resolution. And once we build our cross-modality model, we can then predict into the entire tissue section the high spectral quality of the top example, but we can do that across the entire tissue section at the higher spatial resolution at 30 microns. So this is an example of what we call spatial sharpening, using this predictive capability of these models that we build, and also what we call out of sample prediction, because we have not collected data on the left. And this part of it is really important, because this would effectively be analogous to what we're doing with HubMap in trying to relate data collected from one region of the kidney to another. And just as kind of proof of concept, and you know, as I said, we did not measure on the left, but because we didn't measure there, we could go back and actually measure the ground truth measurements using this high performance platform at 30 microns. So here in the middle, you're seeing the prediction, and here on the right is the actual measurement where we went back just to test ourselves afterwards to see how well things matched up, and they do quite well. We also have a bunch of statistical measures for seeing how well that these types of connections can be made. So that's a brief snapshot of what we're doing at Vanderbilt, and also why I think the common coordinate framework is so critical from a data standpoint and a multimodal imaging standpoint. You know, for us, I think the spatial information is going to be a major link that we use throughout HubMap to tie these measurements together, and it's really establishing this common coordinate framework, in this case for human kidney, but ultimately across all organs, that'll be critical for us making these types of correlations and ultimately leading to deeper discovery as part of HubMap. And with that, that's all I have.
Thank you very much, Jeffrey. And I think we should clap for both of you. Very good. Thank you all. I saw that some people raised their hand, and we have a very unusual setup here. So for instance, Deborah Harayashi raised her hand, but I can't see her in the chat window, so I can't directly chat with her. But I just sent you an email, Deborah, and maybe you have a question for us. And Ellen, right here, she might know how to get you unmuted so that you can ask the question.
I can read Deborah Hoshizaki's question. I can see it.
Oh, oops, I can't from here, sorry.
So her question is the overline based on slices from the same sample, or can it use structures to overlay data from different specimens? That's for Jeff, I think.
Sorry, I just had a very large hawk fly into my window, so I know it's totally random, but I got very distracted there for a second.
So Jeff, can you hear me?
Yes, go ahead.
Because I think I'm now unmuted. So I think that we talk about overlaying data, and we talk about 3D reconstructions.
Sure.
And I just want to make the distinction between having a specimen and you do a series of slices and you and then you do 3D reconstruction of that particular specimen, versus really kind of, I think where the future is going is the ability to take information from slices which are coming from different specimens, but because the ability to identify or mark particular structures, one can then reconstruct, in some senses, a canonical 3D or even a 2D image, which integrates a whole variety of different types of data. And so I just wanted to hear your thoughts about where you guys are in terms of being able to pull data from different samples,
right,
and put stuff together.
Yes. So, I think so. Building the initial data set is the first case. So effectively, it's a single piece of tissue that you're rebuilding a three-dimensional volume of that specimen. But I think I agree with you in that the future, I do believe, is using tools like what we have, but other tools in effectively predicting out of what we call out of sample prediction, so that we can project that data through the entire 3D volume of the kidney in our case, and then also make connections with other data in the process of doing that. So in our case, what we tend to think about is using registration and segmentation to effectively build 3D volumes from a single specimen, and then using machine learning techniques like data-driven image fusion from Roth's group to do this kind of projection of the data through the entire kidney, and then making links with other data sets that way. Did that answer your question, Deborah?
I think that at some point Rahul Satish is going to get involved, because I think he was very keen in making sure that as data was being generated, that there was some thought to giving about what structures or architectural structures, for example, should be highlighted or noted in some fashion, and with the idea of using those architectural structures as a way of reconstructing, doing the overlay with different data types.
Yep. Yeah, and part of that is through segmentation. So we are working on, at least on our side, and I know others as well, is using the multiplex immunofluorescence, and in some cases autofluorescence, to automatically segment and identify substructures and cell types, so that you can also use that information to make connections.
Right, so you can do that. So for GLOMs, for example, you can look at just the information in GLOMs, but when we start thinking about a section of tissue where you may have 6 GLOMs, or is there a way that you can, so this is, in some senses, this is having a roadmap, a physical, car map, and then being able to overlay that Starbucks and all sorts of other things, which may have been generated from a Starbucks list of here are where our
Deb,
Well,
can I
anyways,
jump in? Maybe
I'm going to stop now.
yeah, maybe you can wait until I make my presentation because I have probably exactly what you're talking about.
Okay, thank you.
Are there other questions?
Yeah, I have one. This is Samuel Friedman with OKSI. I'm with the MC Indiana team, but I'm out in Los Angeles. We had just talked about all the different levels of imaging from like some of the RNA stuff all the way up through PET scans. How do we stitch all those different silos together and make sure that we have a clean mapping from one layer up to the other?
Okay, Sam, maybe I can answer something. Can you hear me?
Yes.
Okay, so in the examples that Jeff, so the connection between the different data silos, if the content is not the same, then the content cannot be used to make that connection. So we need to look for some commonality. The commonality in the case of imaging is often the spatial domain. So in the examples that Jeff showed, the connection there between these different heterogeneous data sources was made in the spatial domain using image registration. Now, if you have different, if it's the same animal or even the same section, then the registration is relatively straightforward. If you have the same animal, even if it's different sections, it's still doable. If you have to map between different, pieces of tissue that are from different animals measured at different locations, different humans in this case, I'm thinking animals because of the examples that Jeff showed, but if you're between different samples at different locations, then we need to make that spatial connection via some other way, and that's actually where the CCF comes in. So if the CCF can give us some idea of what is approximately in the same area as another measurement in another technology, then that connection is made again. So you can already feel that as there is more variance introduced by having more distance between the samples or where they were measured and all that stuff, it will become less exact spatially. But as long as there is a measure of proximity, these types of models can still be built.
So my concern here is that as we go from individual to individual, that the spatial component related to the CCF will not necessarily be sufficient to be able to go through all these different scales. And then I also wonder, how do you start connecting the various omics that you would detect or measure and have those also mapped through? Because it might be possible to say, oh, look, I saw something here, it couldn't possibly be that because of the measurement here. And so you then can sort of leapfrog your way through even without having the spatial information that you need. So that's why I'm curious about how the stitching would happen between individuals.
Well, preferably you would want some sort of Cartesian mapping between coordinate systems. That would be the absolute best situation. Now, where that's not possible because there's too much biological variance between the samples being measured at different locations, or there's other types of variables that sort of perturb that. In that case, you still, when I say spatial connection, it doesn't need to be a Cartesian spatial connection where you have an X and a Y coordinate or something like that. It can be something along the lines of, if you recognize certain anatomical structures, I don't know, the medulla or something else, and you can say these measurements come from the medulla, and you have another set of measures that come from another medulla, but it is for sure a medulla. Again, that might already be enough spatial connection to be able to build models. Now, of course, the more precision you have in that, because the medulla is pretty broad statement in terms of anatomy, that would already work. But if you want more precise connections to be made, then the more precision you can sort of infuse the spatial connection with, the farther you get. So anything that is a spatial connection will work, but the more accurate this connection can be made, the more refined the relationships that you can discover.